Okay, let's finish up the antibody section. And remember, 4.9 gets a little bit esoteric. I'll cover it anyway because it's in the chapter and you may be interested in what's going on there. But it's a little more detailed than I can ask for on an exam. All right, so let's talk parts of the antibody. So a variability plot is our vocabulary term. If you were to compare a lot of different antibodies and look at the parts where they're the same and where they are variable, you could plot that as the amount of variability. Does, this particular part of the protein change every time, every second time, every third time, something like that. And so this shows us which parts of antibodies are variable and which parts are not. And you'll see those graphs in just a slide or two. Hypervariable regions are parts of the variable domain that are just extremely, extremely variable, like where any two antibodies may be different. And so each V domain has 3 of these hypervariable regions, which they abbreviate as HV, hypervariable 1, hypervariable 2, hypervariable 3. All right. And yeah, the variable parts are what contributes to the diversity. So what allows them to bind to different antigens. It's the variable parts that make antibodies as powerful as they are. Otherwise, antibodies would probably just be able to bind 1 or 2 things, and then they wouldn't really be much better than toll-like receptors or nod-like receptors, for example. But antibodies are wonderful because of these variable regions, hypervariable regions, rather. These are also sometimes called complementarity determining regions, or CDRs. We're just going to call them hypervariable regions, but if you see them listed as CDRs later on, it means the same thing. It determines whether or not the antigen and the antibody will bind. So it determines whether there will be complementarity there, which would allow binding. All right, framework regions are basically everything around. So remember when I was saying hold up your hand and the fingers are the variable domains? Now you're just going to hold up 3 fingers because there's only 3 variable domains. And the palm is still going to be our framework region. Region 1, 2, 3, and 4 are the FR regions. And the antigen binding site is going to be somewhere at the tip of each arm of the antibody. And it's going to be all the parts of that antibody that actually make physical contact and bind non-covalently to the antigen. So yeah, this could be a very small area, a very large area. Tends to involve something like 15 or 20 amino acids for each antigen binding site. Okay, combinatorial diversity. Yeah, diversity among antigen receptors, like antibodies, for example, that is generated by combining separate units of genetic information. So to make a light chain or a heavy chain, we actually have to combine together several different parts of the gene. And each one of those parts comes from a library where we have them all lined up right in a row in your DNA. We'll have all of the first part, all of the second part, all the third part, all the fourth part. And in order to make a kappa light chain, you will choose from the kappa section one of the first part, one of the second, one of the third, and one of the fourth, and push those together. Yeah. And this is the same process that we use to make antibodies and alpha beta and gamma delta T cell receptors. So this is the one process, this recombination, that drives your adaptive immune system. Shown here, look, it's a plot, and it has variability, oh my gosh, on the y-axis. This is a variability plot. So shown here, residue means which amino acid we're talking about. So we're starting at the beginning of the protein, we're going up to the end of the protein. And you can see that it's not very variable, except in these red regions, it is hyper-variable. And so we've got our framing regions 1, 2, 3, and 4, and our hyper-variable regions 1, 2, and 3. And so that's in the heavy chain variable region, this little bit at the top. And in the light chain variable region, oh my gosh, it's kind of the same thing. because these probably evolved from two copies of the same gene. We just duplicated that heavy chain gene, chopped off the bottom of it, figured out how to put a disulfide linkage in, and boom, yeah, antibodies have been evolved. And so the light chain will also have three hypervariable regions in there. Okay. So yeah, the hypervariable regions actually form loops. And so this is where our hand and fingers are going to come in. So imagine the yellow bit is your palm right here. Imagine the red bits sticking up here are your three fingers. These are the hypervariable domains. And so somewhere, probably involving those fingers, is going to be the antigen binding site. It's going to be a surface that we make just by folding up all those different parts of the antibody. So an antibody is like this three-dimensional block that makes a particular shape, and it has a particular surface with a particular combination of hydrophobic and hydrophilic amino acids on it. And based on that exact shape and charge distribution, it is able to bind to a particular antigen. Okay, monoclonal antibodies are what we use very commonly in the lab. This is where we have basically made a clone of an antibody-producing cell. And so once that cell is committed to an antibody, it can only make that one antibody. We'll see a little bit later, actually I think it might even be in chapter 5, that there's a process where the young B cell or the young T cell will actually chop out all of the other possible genes that could encode for variable chains or constant chains, and it'll throw them away forever, permanently. And so if you can get out a mature antibody, one that has IgD on its surface, that antibody will have one type of, or rather that antibody producing cell will only be able to make one kind of antibody. And if you can capture it in the laboratory and basically farm it, you can make these monoclonal antibodies. And if it's a monoclonal antibody, it will sell for thousands of dollars for just a tiny little amount. So this is a good way to make some money if you understand a bit of immunology. Okay, so this is just looking at the way that an antigen can bind to an antibody. It can bind several different ways. So you can have a little tiny antigen that fits into a pocket. There we go. That's like a little hapten that would bind. You can have a big long protein that slots into a groove at the tip. Great. You can have an antibody that'll kind of dock into this great big sort of shallow depression at the top. Or you can have an antibody that actually has a little lump on it that'll fit into a hole in the antigen. These are just some of the different ways that antibodies and antigens can bind, but just showing you that, yeah, from the same basic structure, by varying these loops in the center and making them bigger, like here, or smaller, like here, or just the same size but changing their composition, you can change what is able to bind to that antibody. All right, more vocabulary. And so it's nice to have these vocabulary after each slide so that you can make your little flashcards and make sure you've got these things down. So an epitope is whatever part of the antigen the antibody actually binds. It's the part that makes contact with the antigen binding region or antigen binding face of an antibody. So these epitopes can take a couple of different forms. They can be conformational, which means it's an epitope that can be made up of, let's say it's a protein here, but it can be made of different parts of a protein. So just like the tip of the antibody is all these different loops, these three different hypervariable loops that come together and make a binding site, they can sometimes bind to something similar, where you've got different loops of a protein that come together to make a particular shape. So those are called conformational epitopes or discontinuous, because we're binding parts of the protein that aren't necessarily right next to each other in that protein. They may be spaced out, and just when the protein is folded up, they come together. Like if you lay your hand out flat and spread out your fingers, and then bring those fingers together in a little point together, you've brought them together and you've made basically a conformational epitope. Let's see. So the other kind of epitope is a linear epitope or a continuous epitope. This is just a chunk, like amino acid 1, 2, 3, 4, 5, 6, 7, and 8 of an protein in a row that form an epitope. And that's fine. Sometimes those epitopes are bound as well. These continuous or linear epitopes are easier to work with in the laboratory because we can do tests that figure out exactly which linear epitope an antibody binds. It's kind of difficult to figure out which conformational epitope an antibody binds in comparison. Okay, how do antibodies bind? Yeah, several ways. One is electrostatic interactions. So this is positively charged atoms on some binding to negatively charged atoms on the other. And they call this thing also a salt bridge. So it's just, plus and minus getting as close as they can together and being locked into position. It's not a covalent bond, though. There are hydrophobic interactions where, because the antibodies are floating around in water, there are going to be some parts that are repelling water, that are hydrophobic, that are sharing electrons in a hydrophobic way. These parts like to bind to other proteins that are the same shape and are also hydrophobic. So two hydrophobic things will be driven together by water, and you'll get a hydrophobic interaction. You can also get pi-cation interactions, for example. And so this is a aromatic ring, like you would find on the amino acid phenylalanine or tryptophan or tyrosine. And so those electrons are going to be in a pi orbital, is what it's called if you remember back to your chemistry. And those can bind to a positive charge. So in some cases, like a actual positively charged ion, like a sodium, or potentially a positively charged part of a protein with an actual positive charge. Included in those, I think, electrostatic interactions, you're going to have your dipole interactions, which are going to be things like hydrogen bonds. And it's mostly hydrogen bonds and hydrophobic interactions that drive antibody binding. But the exact combination is going to be different for every antibody-antigen pair. So here are the non-covalent forces that can hold it together. You can have your electrostatics, positive and negative. Great, okay. And so this is showing a group that would be commonly positive, an amino group, and a common negative group, this carboxyl ion. You can also have hydrogen bonds, where an oxygen is basically got a little bit of a negative charge to it, and it's reaching out and trying to grab onto a hydrogen, but it can't quite steal it because the hydrogen is already bonded to something. And so the oxygen and hydrogen will make a hydrogen bond, which will hold things in place pretty snugly, about minus 1 kcal per mole, if I remember correctly. Then you got your van der Waals forces. These are going to be based on electron clouds, and so positive and negative are going to be attracted to each other and can fall into an equilibrium, which would lead to binding. You'll have your hydrophobic forces, where water molecules are going to drive basically the hydrophobic parts together. And you can have your cation pi. So shown down here, we have our little aromatic ring with all those pi-bonded electrons, or rather pi-valence electrons. And you've got your cation at the top here is showing a sodium ion, but you could also have one of these, for example, I believe. And so, yeah, then you'll get your cation pi interaction. And you can have, I think, possibly multiple cation pi's that hold together some of these proteins. Okay, 4.9, this is the part that's not on the test, but you might as well see it, right? So this is actually looking at a particular antibody and its antigen. So the antigen is going to be a thing called lysozyme. So it's a little protein that's shown here, this yellow bit. And the antibody is going to have the heavy chain variable region is right here. The light chain variable region is right here. And so remember, these have the extra 2 beta strands in their beta sheets. They have 4 and then 5 instead of 4 and then 3, like up in this part. And you can see on the surface, there's a little amino acid, glutamine 121, that happens to reach out and bond to, make little hydrogen bonds here and here. So oxygens are reaching out to hydrogens in both of those cases, and over here off this tyrosine. So each of those dots are a little hydrogen bond. And let's see. Yep, I don't see any other bonds that are driving this particular interaction. But that's just one version. So shown here is another type of interaction that you can get. And so this is a virus. This is the West Nile virus. And shown here, they've chopped off some FABs and put them in with the West Nile virus. And you can see them decorating all over the surface. So all these blue and green little bits here and all the purplish bits over on this side are the FAB of the antibody. So here's the full FAB that's bound. And so this is showing the contact phase. So we're mirroring the structure and the distribution of electrons in such a way that these things will bind. Okay. So there are some interesting versions of these that you find out in nature. These are very useful in the laboratory and in biotechnology. And so just for that reason, it's worth bringing up. So some animals have heavy chain only immunoglobulins. And these are, like I said, just heavy chains that have that variable region at the end so they can bind to whatever the heavy chain can grasp. They just don't have a light chain to help it bind to more stuff. So they're a little bit more limited, but they are definitely around. And things like camels and llamas and alpacas will have heavy chain only IgGs. There are also immunoglobulin new antigen receptors, so IGNARs, yeah, which are a different form of, it's like a very long version of a heavy chain only IgG molecule. And many kinds of sharks, creatures related to sharks, will make these IGNAR type antibodies. And so again, it's just a heavy chain. It's going to be two copies of the heavy chain coming together. To make what is effectively still a working antibody. There are also single chain antibodies, which is a term that we can refer to either of these things, these IGNARs or these heavy chain only antibodies. You'll also see the ones from camels and llamas called nanobodies. That's just like a trendy term that people give them to try and sell them. So if you run into something like that, nanobodies are just another version of a single chain antibody, just meaning that it's only heavy chain that you have to make. These are useful in biotech, like I said, because you can just make one chain and boom, it is ready to go right out of the box. You do not have to have the correct number of heavy and light chains, and they do not have to be correctly bonded to each other. So these are incredibly useful, and you can make a lot of them really quickly and screen through libraries of them to make new antibodies and sell them as a profit or use them in medicine. So shown here for comparison, this is an IgG molecule, and here we've got our light chains, we've got our heavy chains, we've got our disulfide bonds holding the whole thing together. Something like a camel or a llama would have one of these things. So these are the ones they sometimes call nanobodies. And you see it's really just a heavy chain that's bound together. It's just like the blue part, only with one fewer disulfide bond in it. But that's all they make. They do not have light chains. They only have the heavy chains. And so if you can find one of these that actually binds to your antigen, then they're really small, easy to make, easy to encode. You can just put a gene for this into a cell, and it will make its own antibodies, its own nanobodies. The shark version, the shark IGNAR, is just actually just a really long chain. It's got a whole lot of constant region on it, and I'm not sure why that is. Maybe there are more than one protein that has to grab onto the end of this thing. But yeah, you can see the end of it works essentially the same. And so this is another way to make antibodies that can actually are just on one chain and can be a little bit more useful for biotechnology applications. All right, that's the end of our antibody part. I hope you survived all the way to the end of that. And it's not so bad. We're going to have to go, and I think in the next chapter we're going to talk about the recombination that actually brings these parts together. But this is our nice introduction to how things are going to work. And chapter 4 and 5 really are going to go hand in hand. So that's the end of the first part of chapter 4, and then we're going to go to the second part of chapter 4 in the next section. Thank you very much.